Before the 20th century, infectious diseases were the greatest cause of death. The discovery and development of antibiotics seemed to herald an end to these. However, thanks to increasing antibiotic resistance, we are at risk of returning to a pre-antibiotic age, where a mere cough or cut could kill us. On that cheerful note, good evening everyone, I'm Tara, and tonight I hope to present you with a possible solution to our current antibiotic crisis. First, however, we need to start with antibiotics and antibiotic resistance. Antibiotics are substances that kill bacteria or prevent them from growing. They are produced naturally by bacteria and fungi to help outcompete their neighbours and predators. In response, bacteria naturally evolve defence mechanisms to these substances, and this is what we term antibiotic resistance. Human use of antibiotics over the last century has accelerated this process. When we use and overuse antibiotics, we kill normal, non-resistant bacteria, paving the way for resistant bacteria to survive and proliferate. Consequently, infections become more and more difficult to treat. The solution? Bacteriophages, or phages for short. Phages are viruses that infect and kill bacteria. How do they do this? First, a phage will bind to the surface of a bacterial cell, then inject its DNA into the cell, basically hijacking it and forcing it to produce more phages. These phages produce proteins called lysins, which punch holes in the bacterial cell wall and cause the bacterial cell to lyse or burst. New phages are released and they can go on to infect more bacteria. But how can we use viruses to treat disease? Well, phages are really special viruses. They only infect bacterial cells, so they are harmless to our own animal cells. On top of this, viruses have a rapid life cycle, so they act quickly and efficiently against infections. Finally, they can be administered in a whole range of ways. For example, by injection straight into the site of an infection. In 2016, phages saved the life of an 80-year-old man who had suffered for 4 years from an antibiotic-resistant infection. 10 milliliters of a phage and antibiotic solution was injected directly into his infected chest cavity. Within 4 weeks, the infection was completely cleared. A less invasive method involves flushing an infected site with a phage solution. Doctors not far from here in Adelaide found that this method was effective against chronic sinus infections, which is a fairly common problem. In fact, phage therapy worked where normal treatment and surgery had not. Making phage therapy mainstream, however, has been difficult, partly because not enough controlled clinical research has been carried out. On top of this, we're used to using simple chemicals as drugs, not viruses. But phages come with several advantages. One of these key advantages is that they have very high specificity. This means that each type of phage is specific to one species of bacteria. Thus, scientists liken them to guided missiles. In contrast, antibiotics tend to kill indiscriminately, which means they affect even the good bacteria that we need. Others argued that bacteria will evolve phage resistance, like they have to antibiotics. And yes, this is practically guaranteed, and I know that sounds terrifying, but phages are still organisms that evolve in response to bacteria. Imagine the two are engaged in an intense arms race. So we will not have to keep updating phages because the phages will do it themselves. Still nervous about using viruses as medications? Well, the least we can do is adapt their really incredible weaponry. Those lysins that phages produce can be purified and turned into simpler drugs. A 2001 study found that these lysins cleared respiratory tract infections in mice in just 2 hours. As you can see, phages have the potential to be our greatest weapon against antibiotic resistance. Using phages will reduce our reliance on antibiotics, thereby slowing the development of resistance. This will not only extend the lifespan of our current medications, but also buy us time to develop new ones. With phages, bacteria will soon find that resistance is futile.